Hi, my name is Alan Van Deynze, and today I want to talk to you about the African Orphan Crops Consortium with my colleague Deynze and Waweru. To put it in context, last month the 2020 Global Nutrition Report came out with some sobering numbers, where 1 in 9 people are hungry and malnourished, but a more important number we think is 1 in 4 children under 5 are stunted. This also includes young mothers. This is not only a physical condition, but a mental condition and leads to economic hardships. Most people in this situation really don't have a choice and have poor access to a healthy diet and quality nutrition care. If you look at the African continent, about half the countries really have 3 problems where there's overweight, anemia, and stunting, and the other half of the country have anemia and stunting. The Global Nutrition Report goes on to recommend that we build an equitable, resilient, and sustainable food and health system. We focus on nutrition, especially in communities that are at risk, and we focus on joint efforts globally and continue to expand our commitments to improving this condition. It turns out that these are the same goals for the African Orphan Crops Consortium, where we want to reduce stunting due to malnutrition through plant breeding. The idea is to develop genomic tools for key crops in African diets. We chose 101 different crops where we'll sequence the genomes of those crops. We'll also sequence 100 lines for each of those species as the basis of plant breeding programs. The other main contribution for the African Orphan Crops Consortium is the training of 150 of the top plant breeders in Africa to use the latest strategies and technologies in plant breeding. This idea was started by Dr. Howard Yana Shapiro back in 2011. Howard is a fellow at the University of California, Davis. He's also a fellow at the World Agroforestry Center in Nairobi, and he's the past chief ag officer at Mars Incorporated. Generally, the African Orphan Crops Consortium aims to make nutritious crops that are productive, and the benefit will come through plant breeders. So how were the crops chosen? We sent out surveys to anthropologists, sociologists, plant breeders, scientists that are working in Africa, and asked them which of the crops that are already important in African diets in rural Africa. They make up 45 different families, 28 different orders. They include grains, legumes, vegetables, fruits, and about half of them are from tree crops as well. This is to compare to a single order, Poales, that make up most of the calories that we eat in the world with wheat, rice, and maize. So where are we on the genomics program? We've published 8 different genomes so far. We're working on 14 genomes in 2020, and we hope to release those in 2020 or shortly thereafter. Through the Illumina Agricultural Greater Good Initiative, we also sequenced 37 transcriptomes on 37 species, and we're working with 30 additional public programs that are working on some of these crops. All of the information can be found on a single database hosted by the University of Ghent at VIB called ORSE. This is hosted by Dr. Yves van de Peer, who joined us as a partner early on. So the African Plant Breeding Academy is a professional development program. It's a very advanced course in plant breeding, and the participants are actually invited, really professors that are working in the national programs or CG centers in Africa. And the main criteria is that they have to have an active plant breeding program. The idea is that they can take the information home right away and integrate into their programs. The program first reviews genetics, experimental design, statistics, plant breeding, and then moves on to an advanced level to incorporate the latest strategies, genomics, phenotyping, and so on to enhance genetic gains and speed development of improved varieties. These are product-oriented programs that we're teaching that are proven in industry, for example. And we demonstrate with real-life examples, and we bring in experts from all over the world to discuss different strategies. We're empowering African plant breeders. So far, we've graduated 4 different cohorts that make up 112 African scientists from 27 different countries. 87% are PhDs, 38% are women, and they work on 105 different crops, 55 of which are orphan crops. We already have the next class queued up for December 2020, where we've invited 40 different new participants, and about 18 months later, we'll have 20 more people invited for class 6. Together, they've created the African Plant Breeders Association and a community of practice where they can share the latest technologies, the latest developments in plant breeding in their crop, as well as bring in students and educational programs. This is exactly what they've done in October 2019 with their first symposium in Accra, Ghana, where they had 421 people that joined them. The next meeting will be at Makerere University in Uganda in October 2021. Together, they've raised $40 million in grants since they've met each other. This is because they're now writing grants pan-Africa for multiple countries, multiple crops, as opposed to writing grants perhaps for a small crop in their country alone. Since they've graduated, we've tracked that they've published 171 different peer-reviewed articles, and they've released 36 different varieties. The program is run by Dr. Rita Mum. It's coordinated by the University of California, Davis. It's hosted at the World Agroforestry Center in Nairobi, and it's funded largely by Mars Incorporation and also by AGRA and John and Therese Curtis. A green revolution for Africa is unfolding. So who is the African Orphan Crops Consortium? It's truly an uncommon collaboration. The important thing is the African Union development arm is a primary partner from the very beginning. We're very proud that Oxford Nanopore has become a partner in the last year where they're contributing reagents and will put instrumentation in our lab in Nairobi in the next year. With that, I'll turn over the slides to my colleague, Dr. Deynze and Waweru.
Thank you, Alan. So I'm from the Bioinformatics Community of Practice, which was a fellowship that was brought together by the John Innes Center, the Alham Institute, both from the UK, and the Becker Ilri Hub in Nairobi. It brought together 14 fellows from 8 countries and 13 institutions to teach them about bioinformatics for agricultural development. And as a culmination of the work that they did, we were able to study 2 nutritional valuable and climate-resistant legumes, that is Phenostylus stenocarpa, the African yam bean, and Labla papyrus, that's the Labla bean. These 2 crops are very important in terms of food, feed, and nutrition security for Africa. Earlier, Alan mentioned about the high rates of malnutrition and stunting in terms of child development and growth for humans in Africa. And the grains of these 2 crops are quite high in protein, macro and micronutrients in terms of zinc, iron, and other micronutrients, and also in amino acids, leucine and methionine. They are very high in these 2 legumes, and they're not very high in other crops, in other legume crops. And their leaves are very valuable as sources of hay, silage, and forage. And in other parts of Africa and the world, they are also used for human consumption. The tubers of AYB are very rich in protein. So notably, the AYB is able to produce both pods and tubers that are highly rich in protein, higher than other root tuber crops that are utilized within Africa, including cassava, sweet potato, and the Irish potato. They're also adaptable to quite a range of climates and soils because they're able to fix biological nitrogen in the soil. So why the African yam bean? As I said, this is a flagship project of the Bioinformatics Community of Practice, and we wanted to study it because up to date there is no known genome sequence for the African yam bean, and because of that there's very limited understanding into the underlying genetics of particularly why it's able to produce both pods and tubers. And any molecular markers that have been produced up to date have not been successfully mapped to any closely related genome. And so this would be a great resource for plant molecular breeders and plant scientists, not only in Africa but across the globe, to improve on crop improvement. So our goal was to generate an African-led whole genome assembly draft of the African yam bean and be able to release this publicly. And for the Labla bean, this was a collaboration with the Feed and Forage Development Program headed by Chris Jones at Becker Ilri. And we wanted to take advantage of the fact that there's already an existing genome sequence from the AOCC group. They were able to produce a draft genome from Illumina reads, and we wanted to come in and use the long reads and improve the already existing genome. The Moringa oleifera, notably a tree species, which is highly nutritious with edible leaves and pods, is a small shrub that is grown in Asia and Africa as well, and the seed oil is used in the cosmetic industry. It's very highly nutritious. It has high amounts of protein and amino acids like leucine, and then also macro and micronutrients, including the calcium, iron, zinc, vitamin A, among others. So it's a tree species that is very important for nutrition security in Africa. And so for us, this little guy, the ONT mean iron, provided sort of a genomic revolution within Africa. Why? Because number one, it's quite affordable. In most African situations, it's not easy to get a continuous stream of funding, and so you need to be able to take advantage of every little money that you have. And considering that the mean iron is quite an affordable long-range genome sequencing technology, it provides almost a genomic revolution for us. Number two, it's quite scalable, it's very portable. It's being used by scientists all over Africa, and they're being able to do real-time sequencing in the field for both livestock and plant pathogens, and be able to report on quite some novel findings. And then it's also quite easy to procure. For us, it was not a problem at all. Unlike with other technologies, you might have to go through third-party agents. We were easily able to contact Oxford Nanopause Technologies. They provided us with training right here within our labs in Nairobi, and we were also able to get the mean iron quite easily. And so it helps us to circumvent the challenges of limited resources, not only in finances, but also in human capacity, simply because you don't need that much of skills to really operate it. With basic molecular biology skills, we are able to produce quite good DNA. We were able to extract DNA from spin column DNA for the lab, and we're able to get very good sequences. And because we're also able to generate such contiguous assemblies, and also this allows us to go deeper into looking at the challenges that are facing orphan crops, and particularly for the AYB and the lab, we want to look into why they have such long growth periods in the field, to reduce that, of course, for food and nutrition security, and also to understand, for example, for the AYB, how it's able to produce both pods and tubers, and to understand the resource partitioning for that crop. And so for us, during this journey, we learned quite a number of things. Number one, that the amount of sequencing that you do, or the depth of sequencing, is quite important. Notably, if we look here, with about half the amount of data that we had with the lab assembly, we were able to get an assembly that's about 337. This is with the red bin assembler, and the red line here indicates the expected genome size, and we could see that the N50 was quite small. And also with the fly assembler for the nanopore reads, when we use the fly assembler, we still didn't have quite a very long genome sequence here. And also the N50 was still a bit small, about 1,157 KB. But when we doubled that, and we had about 27 gig of data, we could see that even with the red bin assembler, the contiguity of the assembly improved. We had about 354 MB of expected genome size. And then with the fly assembler, we had 369. That is near to what the expected genome size of lab bin is, which is about 437 thereabout. And notably, we could see that we had quite an improved N50 of about 1,442 KB. So coverage and depth of sequencing really affects the continuity of assembly. Another thing that we learned is that it's important to combine sequencing strategies, not only in the type of assembler that you use, but also important to combine both short read to take advantage of the accuracy, and the long read to take advantage of the contiguity. So, for example, here, when we look at the nanopore only assembly, and this is from the African yam bean assemblies, when you used only the Illumina assemblies, we were able to get about just half of the genome size that was expected. The expected genome size for AYB is about 800, and the Illumina only assembly, we were only able to achieve about 487 MB. With the nanopore, we were able to achieve 653 MB, which is is not badly off at all. Of course, we expect longer assemblies from nanopore. It gives us the advantage of contiguity. But then when we are able to combine those assemblies, we are able to see much better results. When we look at also consider the kind of strategy that you use for combining your strategy, this affects contiguity and also affects accuracy. So an Illumina nanopore hybrid here, one of them gave us a 486 MB, which is not good. But then worse off is the fact that the NB is still quite small with this strategy. But then when we also use another strategy for the Illumina nanopore hybrid to combine those two sequencing technologies and use a different assembler, we're able to get 665 MB, which is very good, close to the expected genome size. And the contiguities here are amazing. We have about 402 and 50, and the largest contig here was about 10 MB. So this really improved our assembly. And so we can see this a little clear here with the lab assembly as well, where when you use only the Illumina assembly, you are able to get just about 370, and the N50 here is still quite small. And also with the nanopore assembly only, the N50 is still quite small, and the expected genome size also is not that good. But when we're able to combine those two strategies, you're able to get a very good N50, about 3,142 KB, and then you also have 371 MB, which is close to what is expected. And so this is something that we're also able to notice when you look at the quality of the assembly. For example, here, when we looked at the nanopore-only assembly from the red bin assembler, we're only able to achieve about 67.3% of the genes that are expected in a good quality assembly. Notably, also when you use a nanopore-only hybrid, this is combining the flyer and the red bin assemblers, we're only able to get about 56% of the expected genes that are expected in a good quality genome. But when we combine that assembly, we combine the sequencing strategies and used a good assembler, we were able to get near perfect scores for our BOOSCO completeness. We're able to get about 96% completeness, which means we are getting almost all the expected genes that were expected to be in a good genome. So this was very good. So it's important to also consider the sequencing strategies and consider the kind of assemblers and the strategies that you use to combine the sequencing. For the Moringa oleifera, they went a step further, and they were also able to take advantage of scaffolding from the OmniC proximity ligation from the dovetail genomics. So, and this provided them quite a good advantage. Why? Because if you could see here, the Shasta-only assembly from using only Illumina data, they were able to get 20 contigs, and their N50 was still quite small, 3.8 MB. But when they went ahead to do the Illumina and to do the scaffold, the high-C scaffolding, they were able to get 4 scaffolds representing their N50 and an 18.4 MB N50. And their L90 N90 was still quite good also, 10 scaffolds with an N50 of about 13.8. And this was also reflected in their BOOSCO scores. They had about 98% completeness, 2,075 genes were found with the BOOSCO scores, which indicated quite a good genome assembly. So the AOCC was able to take advantage of the Promethean, so they got quite a lot of data, very high amount of data, and they were able to get good assemblies as well. So for us going forward, the next steps would be to take advantage of the RNA sequencing that we are doing currently, that's going on, to be able to generate transcriptome mapping, to be able to understand and inform on annotation and gene models, and understand a little bit better about the underlying genetics for these orphan crops. And also for the lab and the AYB assembly, we expect that soon enough we'll be able to complete the sequencing of the scaffolding, so that we can be able to come up with chromosome scale genome assemblies. And we've learned quite a little bit, as we've seen, that spin column DNA is also a viable source of DNA for nanopore sequencing. The coverage and the depth of sequencing is quite important for nanopore, as well as combining the strategies and combining both the sequencing strategies and different assembly strategies to improve on your quality. And notably, capacity building and collaboration is very, very important. It has played a big role for us in being able to achieve what we are achieving currently, because all these fellows were trained, they went back to their home institutions, and they are providing the much needed bioinformatic capacity in terms of data analysis and high quality research, because you're also able to bring in the networks that we got from the fellowship. So this is very important, and going forth is going to play a big part in ensuring that Africa is able to play a big role in fighting food insecurity, to be able to increase productivity so that we achieve food security and nutrition security within Africa. And we'd like to thank all our collaborators from the World Agroforestry Center and UC David for the AOCC, and all our collaborators for the Bioinformatics Community of Practice, our mentors, our collaborators from Alham, JIC, Becca, and around the world. These are the funders who are able to fund the work for the Bioinformatics Community of Practice, and we are very grateful to be able to present our work at the Oxford Nanopore Technologies platform in the London Calling Conference. Thank you very much, and for now, we'd like to take any questions that you have. Thank you.